Hi, this is Dr. Ben Morrill. Welcome to episode 36 of Reptile Genetics Weekly. Happy to have you here, and we even have a new intro and outro for you. So we're always looking forward to trying new things. So fun stuff. So for today, the sample update is there's probably lots of you watching this waiting for results because the run is going right now as I'm recording this. And if you watch this, this is Wednesday. If you watch this on Wednesday night when it comes out, the run is literally going right now. So I'll have results coming off this weekend. If the analyzing goes fast, I could have some of it going out on Friday, but at least sometime over the weekend, I should be able to have that. So we'll have a whole bunch of results for people. And we've got a lot of other cool stuff on this run that we're going to be learning, and we'll talk about that a little bit in the episode. And maybe we'll just go ahead and jump in and bring Kayla on. How are you doing, Kayla?
Hey, Ben, I'm doing great.
Good.
Yeah. How about you?
It's been a crazy last couple of weeks, but it's been good. And we'll talk about some of the reasons why, but
yeah.
It's been good.
Well, good. Well, let's get into it. First up, tell us about the puzzle updates.
Yeah, so for those of you interested in puzzle and GCR axanthic, both of those, I have a bunch of samples on this run that will have results this weekend.
Oh, yeah.
Yes. So as long as that goes well and they're clean, then we should be good to go to, as far as on our end, make those available to customers. And we're talking to Clutch and Morph Market about getting those added. And like we've talked about before, the biggest thing we need right now is to make it so that the panel stays what it is right now. So any new tests like puzzle and GCR will not be part of the panel for a little while. So that will give me now that like I said, we've talked about this before, but just for people that didn't see before, now that we have so many tests in the panel, each time we add another one, it takes me a lot longer. It's kind of like an exponentially more complicated thing, because before I only had 10 other things I had to get to go well with the new one. Now I've got 22 or 25 or whatever. So we're going to have the panel be fixed at, I think, at 22. And then puzzle and GCR won't be on it. The others that we're ready to go with are the Blue-Eyed Lucy tests. So we'll have Special and Mojave and Bamboo and Butter and Lesser and Mystic and Phantom, all that stuff. But we're not ready to do that yet because right now, if we get them added, then they're going to automatically be part of the panel. And I can't do that yet. I can't make it work in the panel yet. So
we can't have your head exploding. We need you.
Yes.
Yeah. Okay. Fair enough.
As soon as we get those updates in Clutch and Morph Market, then we'll have a whole bunch. We'll have like 8 new tests we can add. So that's pretty exciting. Yeah. We'll officially be to 30 that will be available, 30 or more that will be available for customers. So, and then, like I said, the panel will stay as it is now. I think the newest tests on there are Orange Dream and Leopard, if I remember right. So we'll include that. So
sweet. All right. And so as far as expediting tests, I know you were talking about making some a little bit faster.
Yes. So that's a big part of why I've been so busy and I am so tired. And I did get a little sick. I have a little bit of a cold.
Oh, I'm sorry. So, hope you feel better soon.
But yeah, so it's been a whirlwind. We want to be able to now we've got to the 30 once, we are able to add puzzle and GCR as well. We wanted to focus on getting the tests faster and cheaper if possible. And we talked a few episodes back about some potential new technology we may be able to use for shed testing. And we have officially gotten it to work with Desert Ghost, specifically DGC Clown and VPI axanthic, which this animal is a beautiful animal that happens to have all 3 of those. So
yeah.
And actually it was one of the animals I used to validate all 3 of them because I could test all 3
really?
from the shed from that one animal. So
that's perfect.
Yeah. So the awesome thing about this, it's really going to be a game changer. As we'll try to transition more of the tests over to this fast technology. But what it means is when your test comes, like right now, your shed gets to us and you've got about 3, maybe 4 weeks until you're going to get your results. Well, with this new fast technology, which, like I said, it's working, but now we have to, set it up so we can do, the high throughput with it. So it's still going to be, 2 or 3 or 4 weeks or something for us to get it so that we can crank these out. But it will get to where your shed gets to us and will be pretty easy for us to have your results back to you within 48 to 72 hours. And a lot of the time it'll be within 24 hours.
Yeah. Oh, that's going to be so cool.
Completely different. Yes. The quality is just as high. We're getting the same, over 99% accurate. Reruns are lower. I'd say, I think statistically they are lower. But I mean, it's pretty similar to the number of reruns that we have in general with the sequencing testing we've been doing. So we won't really lose any quality or anything, but it'll be faster. And it will also allow us to make the tests cheaper. So for tests that we've had around for a while, like Clown, that one will be significant. Like it won't be $65 anymore once we can switch it and run everything through the fast test.
Awesome.
It's going to be significantly cheaper. And then on top of that, like we've told you all, in January and February especially, we'll have deeper discounts. So if you're getting 10 or more animals, that'll get to where you're going to be able to get discounts on top of the lower price for Clown. You're also going to get, if you're doing 10 or more at a time, you're going to get significant. We'd have to ask Sean what it's going to end up being, but it'll be more than 10% off. It might even be up to 20% off. So it's going to be faster and cheaper. For newer tests, especially brand new ones like Puzzle, like if we get Puzzle and GCR onto the new technology, you'll get your results faster. Those will still be at that, $60, $65 price for a little while, just to help cover the research and development costs to be able to make these tests. But once we've had them, a test has been around for, some amount of time, we'll have to see what will help cover those costs, but, matter of weeks or months. Then those will also drop. But everything, regardless of what price it is and whether it's fast or not, everything will be quantity discount now. You'll be able to get a quantity discount. So,
well, cool.
Making lots of progress, new tests and faster. And it's exciting. I especially, the thing that's been the most stressful to me, because I want to provide these tests for people that, are breeders like me.
Yeah.
I hate that it takes so much time. So being able to move towards this, where we have your turnaround times in 1, 2, 3 days instead of 3, 4 weeks, is going to be a big load off my mind. I'll be much more excited to help customers, talk to customers when I know we can churn this fast. So I'm excited for that.
Absolutely. Well, awesome. So I hear you've been on some podcasts.
Yeah. I got to be one of the 3 wise men here.
I still can't get over how beautiful this is with and Travis. We've had him several times on the show now. So having him just sitting there in a manger is hilarious. Like I just admire it. It's great.
He gets to be the savior. So
yes. So what you guys talk about?
It was a good podcast. The other thing, if you haven't been on the Reptile Gumbo podcast, their Facebook or Instagram, he also made a really good short video where it's our faces, on other people's bodies, and it just says doctor.
I downloaded it just for this.
Oh, did you? Right on. You have to play it.
1 sec.
So that was the interesting thing was he was able to get 4 doctors to come on the show at the same time.
The all doctors show.
Yeah, they're all into genetics and into keeping reptiles. So it was a pretty interesting conversation. It was live last night, and then I think the recorded version he puts up like in 5 or 6 days or something like that. So so those of you that caught it live, you got to hear it right away, and those of you that want to find it, I think it'll be a few days until it's posted. I know it goes up on YouTube. It might go some other places too.
Good stuff. Just got the video. Here it is.
All right.
Doctor.
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 So he did a great job preparing for that. It's so funny. Great posts that he did.
I don't know what clip, like what movie or show that's clipped from, but it's
I don't know either, but it's great.
Yeah. All right. So you mentioned another thing you talked about in that podcast, right?
Yeah. So on that podcast, one of the other doctors is Dr. Warren Booth, and we were talking a little bit about grain tree python and specifically a sex determination test. And I mentioned, sorry,
You're good.
I mentioned that it's one that we've been working on for, it seems like an eternity now.
It's like our holy grail, you know?
Yeah. To be able to get a single shed test like we have for colubrids and like we have for ball pythons to do sex determination for green trees. That's literally the number one thing, the reason why Sean and I wanted to start a business like this. And when we talked about it back in 2011, that's like the one test we wanted the most. And it's the one that we haven't been able to get still to date.
Yeah.
So, and I said, you know, I know Warren, you've looked at this as well. And he said that he can't give very many details yet, which I would take to mean they're going to publish something about this fairly soon. But he said there's something really weird about Morellia and the sex chromosomes in Morellia. So that would include carpet pythons. And so anyway, there's something funky going on. He said that it's unlikely that we're going to find like a specific marker that will work across many individuals. I've been trying to make it work. You know, I'll get it to work in 5 or 10 or 20. But then when I blow it out and do 60 that I know they're males and females, then it always fails. And I don't want to make a test available that doesn't work on a large, you know, at least on 60 different individuals. So that's why we haven't had it. But sounds like there's a good reason why. So unfortunately, it's kind of some bad news. But you know, I'll still keep my fingers crossed and hope that, you know, once they publish that, we'll be able to dig in there and find something that'll work. But we might have to go back to what I used to be doing, which did work perfectly, but only for some pairings.
Only for some, because I know you were testing like a whole, like basically a family tree, right?
Yeah. So what I was doing is I would find a genetic marker on what I knew was part of the sex chromosome in the sire. So let's say that for a clutch of green trees, you sent me the shed from the sire and from the dam. And so I can see, okay, what genetic markers are on their chromosomes. I find one that only the sire has.
Yeah.
And then I get either his dad, so the paternal grandsire to the clutch, or one of that same sire's male offsprings that I know for sure is a male. And then I can see, okay, that marker shows up in his son. And so any offspring from him that get that marker, it had to come from him because the dam doesn't have it. And he's only passing it to his male offspring. And if they don't get it, then I know that it's a female. And so that gave me a way. It was difficult. It's expensive and takes quite a bit of data analysis time. And I always thought we could get a single shed test and not have to deal with it, you know?
Yeah. But now, it looks like it's going to,
Yeah, we might have to go back to doing it that way, which is going to be expensive. You know, it's not going to be something. Well, first of all, a lot of people, like if you just have a green tree python that you got, you know, from a show or from a breeder, you know, you might not have access to the parent and then both parents and either a known male offspring or the paternal grandsire. So it's complicated and messy. And that's why when we scaled up and started to run things, you know, high throughput where we're, you know, running lots and lots of samples, I couldn't really do that anymore. But if we know for sure that we're not going to have a single shed test anytime soon, you know, maybe we need to figure out price wise what that would cost to do it that way. And it's going to be a fair amount more expensive than ball python bluebird because it's way more involved. But it would be nice to make something available that I know works. And, a lot of people probably are going to say that's too expensive, not worth it, but
right.
it's going to be what it's going to be. Like I can't make it any cheap. It won't be as cheap. The little bit I did it 2 or 3 years ago, I won't be able to do it that cheap. So
probably because you were eating a lot of the costs to help, since it was helping you research, right? Or
yeah. I mean, I was essentially putting in lots of hours doing analysis for free.
Yeah.
And now I have so many other things to design and stuff that I have to, essentially I have to charge for the time that I'm spending on that. Otherwise I need to be doing something that, people are paying for. I can't donate my time anymore like I was back then.
Makes sense.
Because there's lots of things that we need to be working on and moving forward. So yeah, and it is different because our process is different now, and so it's not as easy for me to do it the way that I was doing it before. That's part of it too. So it's complicated and messy. That's why we decided that it's just not going to work to make it available. But that might be the only option for the next little while, and maybe forever. Like Warren said, maybe it'll end up that there isn't a consistent marker across all individuals like there is all pythons and all the colubrids that we've tested, except for the only weird one is tricolored hogs. Those don't seem to be consistent. Some of the time the test works clean, and then other times someone will send some in and it just doesn't make sense what I'm getting.
Weird.
They'll send me 4 or 5, and then they'll send me the sire and dam, and it doesn't match up. So
that's wild. I don't know. You'd think that, I don't know, if there was like a with the Morelia, you'd think if there was a centralized like stud book like they do with the AKC or with horses and stuff, maybe you could map out like the most common markers that you do find across that. But as decentralized as the reptile hobby is, I feel like that would be a massive undertaking.
Well, actually, green trees, out of all the pythons, they're the ones that do lineage tracking the best that I've seen. Yeah, they actually do a very good job. Some of these animals are still $5,000 to $10,000 animals. Like,
that's fair. So
they definitely know.
Yeah.
Like, you talk to someone like that animal right there, I bet there's someone watching this that could tell you, even though this is just coming off of Wikipedia, they could probably tell you who took that picture, and then they would be able to tell you who the sire and dam were to that. Like, it's a lot different, especially than like a ball python breeder that's more focused on the morphs.
Of course.
Than like ancestry. But with green trees, they keep up with it pretty good.
I really respect that, actually. That's awesome.
Yeah, you'll see, like, when people sell green tree pythons, you're much more likely to get some kind of a pedigree that goes back a couple of generations.
Yeah.
When you buy a baby green tree from a green tree ball python breeder, captive breeder, you very well could get a little, family tree when you buy that animal.
Oh, I like that. That's cool.
And I've done it some when I've sold ball pythons, but people just didn't seem to care. I certainly keep track. I know where all my animals came from, and as long as I know the animal number, I can tell you all kinds of stuff about them and their history.
Yeah. So moving right along, we have you're going to be in another podcast tomorrow, right?
Yep, tomorrow night. So in the top right there, that's Bill Stiegel. And so that means we're going to be talking about green trees again.
Hey, awesome.
Yeah, so I'll get to pass on this news again tomorrow night. And those of you that already saw this and end up watching MJ's podcast tomorrow night, you already know the bad news. But we'll probably talk more in depth in that episode about the possibility of making that green tree, that messy, complicated green tree python sex determination test.
Yes.
Making that available. We'll have to have kind of two different things. There will be a cost to be able to validate a pair that we can test their offspring. And once we validated a pair, then there would be like a price per animal to do the sex determination. And if you ever wanted to use that same sire with a different female, there'd be a lower price to just validate with her. You just send that second dam, send her shed in. We can make sure that with the markers that the sire has and the markers that the dam has, that we'll still be able to tell you from their offspring.
Okay, that's good.
And have to point out in this, give special thanks to Sean for, so when he was here, or no, it was at Tinley. That's what it was. When we were at Tinley, he's like, hey, I got to get a picture of you with your crazy hair to send to my fiance.
You do look a little more feral in that without your hat there.
So I was like, okay, we can do that. So when MJ asked for a picture of Sean, he sent the picture that included my crazy scientist hair. So
dang it.
Okay,
how opposed are you to me using that picture as a meme later? It won't be too bad, I promise.
You know me well enough by now. I'm not too worried about stuff like that.
Sweet.
That's fine with me.
All right.
But yeah, any of you that are interested in the talk tomorrow, we'll talk about genetic testing and quite a bit, I'm sure. We'll talk about green trees quite a bit, I'm sure.
Absolutely.
So it should be a fun conversation. MJ does a great job on there. So should be a good podcast. And that's tomorrow night, 9 p.m. Eastern is when that one starts.
Yep. Well, definitely be sure to check it out. Let's see. We have some news in the colubrid world. There was a study published recently about how not only how garter snakes make friends, but how they actually organize their society around females. Let me throw up the. Here we go. This is the an article about it in at science.org, but they apparently have little communities, and it's basically building on some experiments that were done with captive garter snakes in 2020, which I thought was adorable because they had like little friend groups and stuff. But apparently they actually do network with like older females. Tend to be the ones that other females will like prefer to hang out with, whereas the males are a little less social.
Interesting.
Yeah. So it's worth a read. The article's cool. I'll link it in the I'll be sure to link it in the down below. But you know, if you like garter snakes, if you like learning about social behaviors in snakes, it's pretty cool. Because like it's important not to anthropomorphize, whatever that word is, to think about your snakes too much as people. But at the same time, we sometimes as reptile keepers push too hard in the other direction and say that no, they're completely not social animals at all. So I think it's cool to learn about how they interact in the wild, and maybe learning about how we can apply that to better husbandry in captivity. So I hope there are more studies done like this because it's cool. Actually, it's time for Why I Do This.
Right on.
Okay, so this time around, we have a. Here, let me pull up a picture just so you can see him. This is an old picture of Tim Spuckler, who is our Why I Do This feature for today. He's there holding a big old timber rattlesnake in West Virginia back in, I think, 2006. And I'm friends with him on Facebook. And he is just such a prolific herper, actually.
That takes excellent pictures.
Yes. He's cool. And he sent us a little write-up, so I will read that for you guys now. Grab my mic. So Tim Spuckler says, I've always been interested in cool creatures. As a kid, I grew up in Cleveland, Ohio, and would collect and keep insects and other invertebrates as pets. It's a pastime that I still enjoy today. Eventually, a family member presented me with a golden nature guide to familiar American insects. I was fascinated at all the neat six-legged aliens that I had yet to encounter. I still remember specific passages from that book. When I was playing behind a friend's garage, I flipped a rock and found a redback salamander, my first wild herp. That was my first wild herp too, actually. And he was hooked. How could something so amazing be living behind someone's garage? And what more was out there? It wasn't long after that I was given from the same Golden Guide series of books, Reptiles and Amphibians, a guide to familiar American species. I was blown away. Not only were the illustrations awesome, featuring paintings of herps in their natural habitats, but to someone single digits in age, the species accounts were astonishing. For example, the hognose snake is unique and amusing. When molested, it hisses, spreads, and strikes, as though to appear dangerous, but it never bites. If threats fail, it rolls over and plays dead. I'd never heard of such a thing before. The book was filled with novelties like alligator snapping turtles, collared lizards, mud snakes, tailed toads, and mud puppies, all unique and interesting in their own way. Reading about a yellow snake that could grow to be 7 foot long that resided here in the United States was an eye-opener. At this time, it was the mid-1970s. Snakes were not as widely available in pet stores, and the captive breeding of them was just beginning. I knew that I wanted to seek out all herps in my golden guide, and have spent much of my life doing just that. I learned that I could ride my bike to vacant lots in Cleveland and find Decay's brown snakes, a thrilling experience. My family moved to a suburb when I was 14, and there were new herps to be found. Milk snakes, garter snakes, snapping turtles, wood frogs, and more. As time went on, I sought out more strange and wonderful beings, and just this year, I found my first ever ringed salamander and prairie king snake. In high school, I discovered the book Snakes, the Keeper and the Kept by Carl Caulfield. On the topic of keeping snakes as pets, he wrote, "The original impulse exists in many of us as an urge to capture and hold beauty, and incidentally, to learn." This struck a chord with me. I wanted herps, and more specifically snakes, in my life every day. At that time, I would keep wild-caught snakes for a while, releasing them back where I found them. In 1980, I joined a herp society, the Northern Ohio Association of Herpetologists, otherwise known as NOAA, and found other people with similar interests. The monthly meetings not only featured presentations, but members also often brought in their herp pets. I still remember the first time someone presented a Pueblan milk snake at a meeting, a species that was only discovered a few years earlier. I'd never seen anything like it. There was no internet in the early 1980s. Not long after that, a striking orange and black snake was available at a local exotic pet store. It was my first encounter with a Honduran milk snake. $300 was a lot of money for a teenager back then, but I bought it. The snake lived 27 years in my care.
Wow, that's awesome.
Right. After keeping snakes as pets for 10 years, the idea of breeding them seemed like a fun prospect. With a resource like NOAA, I could learn how to fine-tune the aspects of breeding reptiles and raising their offspring. In the late 1980s, I produced Honduran milk snakes, Pueblan milks, and southeastern five-line skinks. Cool. It's hard to describe the excitement of seeing reptile eggs hatching or looking into a garter snake enclosure and seeing it filled with newborn baby snakes. The enjoyment that came from producing a few reptiles eventually caused me to expand to corn snakes, king snakes, rat snakes, and a number of other North American colubrids. The art of selective breeding has been a fun path to pursue as well. Interesting and out-of-the-norm colors and patterns of snakes offer genetic pathways to investigate. High black and high white are just two variations of Honduran milks that I'm currently working on. Hopefully another project of mine, the world's first ghost Baird's rat snake, will pan out in 2024. Oh, cool. On the other hand, finding herps in the wild has caused me to really appreciate not only their lifestyles, but also the geographic variations of wild snakes. I continue to be enamored by many of the classic snakes in my golden guides, no matter how common they may be in the wild or in the pet trade. I have a nice collection of yellow rat snakes, and I'm still amazed that the 7-foot, mostly yellow serpent can be found right here in the US, and I can keep them in my home and enjoy them. Yeah!
Awesome.
I love that, Tim. That was great. No notes. I agree.
Yes.
Let's see. I especially agree with the part about, there's a 7-foot big old yellow snake that lives right here in the US, and I can find them, well, not in my backyard here, but I can find them here in the US, and likewise 7-foot black rat snakes out in my backyard. I love that. I think that's the appeal for a lot of colubrid, or at least North American colubrid keepers. And yeah, hard. I relate to that. So here's some photos that Tim sent us. So this was a picture of one of his first captive breeding successes, Honduran milk snakes in the late 1980s.
Those are pretty.
Yeah. And this is his snake breeding setup back in 2006. Again, there's the timber rattlesnake picture, which I love that like chocolate and yellow phenotype. You don't see it often, but it's really pretty.
Pretty.
Yeah. Let's see. Here's an example of, well, he's been producing Hondurans for over 30 years now, and he's produced some neat stuff like these spotted Hondurans, which are really cute. I like the spots. There's a high white albino from his high white project. Let's see. This was an odd looking Nelson's milk snake that hatched with him in 2005. Just look at how black that tail is.
And that head stamp on that looks like a, I don't know, like a dragon or something.
Yeah.
That's awesome.
Right. Let's see. We have an albino Nelson's milk snake that hatched in 2007. That's a nice, big, healthy individual. So pretty. A picture of the first ever pairing of an anarthristic Baird's rat snake with a hypomelanistic Baird's rat snake in 2021. So looks like this is 2024 is probably the year that he's gonna hopefully create a world first ghost, which I'm excited to see. Baird's rat snakes have the coolest subtle colors in between their scales. Like, they're so pretty. And he says in recent years, his main interest has become locality specific animals. So we have Kunashir Island Japanese rat snakes with some eggs behind her. So cool looking. We have some, yeah. We have some Victorville, California locality Cali Kings. Locality Cali Kings. Let's say that 5 times fast. We have San Mateo County, California coast garter snakes. Oh, what a good mama. Some Palm Beach County yellow rat snakes, which love yellow rat snakes so much. And finally, Marin County, California red-sided garter snakes.
Pretty.
Yeah, they're like a more muted version of like normally you hear about the San Francisco red garters, but that's like the Marin County is more muted. I like that. And that is why Tim Spuckler does this.
That's awesome. And any of you that aren't following him, if you're on social media at all, I think he's on both Facebook and Instagram, isn't he?
He's on Facebook. I don't know if he's on Instagram, but
Maybe
I'll be sure to post his social media in the doobly-doo. So for y'all who want to follow his work, definitely it's worth a shot.
And you said he usually posts pretty much every day, right?
Oh yeah, every day, if not multiple times.
Beautiful pictures.
Yes, highly recommend it. So thanks again, Tim, for that awesome write-up and all the pictures, and thank you for posting every day. It makes me happy. All right, and if you want to add in your own Why I Do This story, go ahead and send us an email at raregeneticsocial@gmail.com. We would love to hear your story in write-up or video form. It can be shorter, it can be longer. We like to remind ourselves, why we do this. So if you're having a rough day, you can always go back to these happy moments.
Yes.
So that's all we got for today. Nice 30-minute episode for us.
That's it. Yes. Yeah. Thanks for joining us. Please do share and like and subscribe. Give us comments. Definitely keep Kayla busy with these Why I Do This. Send them in. Give her lots to get set up. We love hearing these stories.
All right. Well, we'll call that a wrap. And thanks everybody for tuning in. We'll see you next week.
New outro
time. Let's go. Okay. New outro in 3, 2, 1.